Welcome back. We're going to take a look in the OpenStax microbiology chapter 14, and we're going to take a look at some antimicrobial drugs that are used for chemotherapy. We'll go through some different examples of natural and synthetic drugs. Take a look at some different things that you want to consider when you're looking at using these antimicrobial drugs in terms of their therapeutic good and bad sides. We'll take a look at how to test the effectiveness of these antimicrobial drugs and take a look at two really important indicators of the concentration necessary for inhibition but also for killing. And we'll spend a huge amount of time looking at different types of antimicrobials, from the antibacterials to the antifungals, the antiprotozoan drugs, and some of the Helminth drugs, and end up looking at some of the antiviral drugs that are out there. And then we will cap everything off with how drug resistance can develop in the populations of these organisms that are out there. It's key to remember that when we're looking at this idea of antimicrobial drugs, we're really looking at chemotherapy, which is the use of a chemical to treat a disease. And this is something that a guy called Paul Ehrlich came up with, this idea in the early 1900s, where he proposed finding what he called a magic bullet, a chemical that could be used to kill the pathogens that are causing diseases without actually killing the patient. Chemists were looking at this and trying to come up with a lot of things, but the first really successful use of a naturally produced kind of chemotherapy drug that could be used for this was actually a accident that happened with a guy named Alexander Fleming. In the 1920s, he was working in a lab in England, and he was actually working with Staph aureus, our gram-positive bacteria that we find on the skin. And he had apparently gone away for the weekend, and he came back and he found a mold growing on his plate, as shown in this example right here. And instead of just throwing it away, he actually took a look at it, and he noticed that there was this zone of inhibition around the mold growth where the bacteria was actually inhibited from growing. And when he thought about that, he realized that the mold must be producing and releasing a chemical that was inhibiting the growth of the bacteria. And this turns out to have been a penicillium that was producing what we now call penicillin. And he spent years trying to purify this to be able to get it to work. Unsuccessfully, I might add, but it wasn't really until World War II that people spent more time and money and was invested to find a way of getting enough of this drug to actually use it to treat injured people, because if you look at the wars that were out there, more people died of infectious diseases. And when you're looking at getting shot by something that can allow these microbes to get under your skin, and the penicillin was used to treat these. And the original drugs that were used were actually injected. They were not given in an oral form because they weren't stable in stomach acids. And there are some stories that I've read where they actually would take the urine from patients who received an injection of penicillin, and they would recrystallize the drug out of their urine, because most of the drug actually was filtered out of the bloodstream by your kidneys, and then that drug could be readministered to a new patient. If we look out there at the different kinds of microbes that are out there, people realized that they could start looking for different kinds of microbes, and they found a whole battery of different kinds of chemicals that could be used to kill off different kinds of things that are out there. And the three primary things that people have used to look for the different kinds of drug-producing microbes that are out there is some gram-positive bacteria that are known as actinomycetes, and these guys actually grow as filamentous cells, very much like molds. And you can see we get a variety of different kinds of antibiotics out of these gram-positives. The gram-positive rods or bacilli also produce some different types of antibiotics, and of course are fungi that are out there. And so it's kind of interesting. If you look, all these guys that are out there are actually spore producers, and so they think that these can survive the effects of the chemicals because they can produce spores, and the spores are metabolically inactive, so they are insensitive to the chemicals, and this way you could kill off your competition, and then the spores can germinate, and the new thing growing out of there could potentially kill the or eat the leftover remains of what's there. This doesn't mean that antibiotics are going to work against everything. In fact, there are some kinds of microbes that you can actually grow in media containing water with antibiotics. So in that case, the antibiotic drug is actually acting as a carbon and energy source for the microbe. These are natural antibiotics. These are drugs that are chemicals that are found in nature, produced by these particular kinds of microbes. The other chemicals that people were looking at were what are known as synthetic antibiotics, and these were also discovered in the early 1900s. And they are completely synthesized in a chemistry lab, and they're not found in nature. But imagine going to get an antibiotic that you have to inject. It's not going to be the best kind of treatment that you want. So people have made what are known as semi-synthetic antibiotics, which are basically chemically modified derivatives of these natural ones. So you take something like streptomycin and you change some of the functional groups on it, or change penicillin so that now it can survive through the stomach acids and go on to still be an active drug when it hits your intestines and gets absorbed and travels throughout your body and your bloodstream. So the semi-synthetic antibiotics are basically designed to change the targets, maybe allow them to affect gram-positives and gram-negatives, make them more stable, change them so that they don't cause a lot of toxicity to the host cells, or a variety of other kind of properties that are out there. When we consider these changes that you want to have for making semi-synthetic antibiotics, you do have to consider a couple of different factors that are going to be associated with your chemotherapy drugs that are out there. And one of them that's really important is this idea of spectrum of activity. Do you want something that is broad range in its activity, like these guys here that you can see they affect gram-positives, they affect gram-negatives, and these little chlamydias, which are actually intracellular bacteria that will replicate inside of your cells. You can see that streptomycin and isoniazid over here can actually affect gram-negatives and our gram-positive mycobacteria. And so these guys are broad spectrum. They will be effective against a lot of different types of microbes. The azoles you can see here, these affect a lot of different kinds of eukaryotic ones. But notice your viruses are in a category all by themselves over there. And when you look at some of these things, these are known as narrow spectrum drugs because they're effective against a subset of organisms. And when you think about this idea, if you're giving a drug to somebody that is broad spectrum, it has a chance of, yeah, it will kill off, say, a bacterial infection, but what if you get rid of gram-positives and gram-negatives? You need to have some of these microbes in your intestines in order to allow you to continue making certain vitamins. They help you with digestion. And the problem with broad spectrum is they can't allow secondary infections to develop. So you take a broad spectrum antibiotic to get rid of a bacterial infection, and you end up with a yeast infection as a result of that. So normally your microbiota that are on and inside of your body are keeping these pathogens in check. The broad spectrum kills off the non-resistant cells that are part of your normal complement of bacteria, and then you get your secondary infection, or you can get a super infection from drug-resistant pathogens that are growing out of control. So whenever you're looking at antimicrobial drugs, you do have to consider this idea of the spectrum of activity. Is it broad or narrow? And the more narrow you can get, the more you can target specific kinds of things. And I often use the analogy of these guys that are broad spectrum are using a machine gun, and you're going to cause some damage to the innocent bystanders. But if you have a narrow spectrum, now your machine gun has got a scope on it, so you can aim it a little bit more effectively. Another thing that you want to consider is the route of drug administration, because that will affect the concentration of it in the blood. And if we look at this little graph over here, what we can see is if you take something orally or you get an intramuscular injection, you're going to see this nice boost in the amount of drug that is able to eventually diffuse into your bloodstream there. And you can see it plateaus and then it goes down over time here. So you are going to see a massive amount or a massive dose of this at the beginning, and then it slowly tapers off. But if you are given something intravenously, you're going to get a massive dose of that drug. And the reason we have to be careful with this is you have to consider toxicity, and there are certain levels that are permissible with drugs that are out there that we want to reach. That we don't want to go over, because then you could possibly cause damage to the patient. So you would want to monitor, especially an IV drug, because it's going to hit the bloodstream so quickly and you'll have a massive dose there. The other thing that we have here is something that is known as the half-life of the drug. And that's basically when you administer your drug, it's going to be around for a certain amount of time. So we start out as, we call it 100% at the beginning, and then in one half-life, you've lost half of your concentration. And then after 2 half-lives, you've lost your 50% and then half of that 50%. So you are steadily decreasing the amount of drug that's there. And so when we look at this, IV drugs, you need to be careful because they are going to peak quickly and then they're going to gradually decrease, whereas these other methods of introducing the drug are going to take a longer time to peak in your bloodstream there. But when we look at antimicrobials, you really want something that is readily available and it's cheap. You want something that's going to be chemically stable, so think I can keep it in my bathroom and it's not going to break down. I want something that's easily administered, so something that you don't have to inject would be great for people to have, but also you want to have something that is not toxic and it doesn't produce a lot of allergic responses in your patients. The other kind of interesting thing when we look at it is another factor that you have to consider is drugs that are out there can actually interact with other drugs. And if you go on to nursing school, one of the things you will get exposed to in pharmacology class is what kinds of drugs can be used in combination. And if you look at this plate right here, we have different drugs that are impregnated in our paper discs right here, and the drug is diffusing out, and you can see this zone of inhibition around here, and then these are the lawn of cells that were plated on this culture medium. Well, within this zone of inhibition, the drug is at a high enough concentration to interfere with the growth of that. But if you notice, this is getting lower and lower in concentration the further you get from it. But notice right here, in this little region, both of these drugs are in lower concentration, but you can see they're both interfering with the growth of the cells, where over here it's not interfering. And what's going on here is this synergistic interaction. And both of the drugs together are more effective than either one is by itself. And so if you have synergistic drug interactions, you can actually use a lower dose and still have it be effective at treating your microbe that's out there. You can also run into something that is the opposite of synergism, and that's antagonistic interactions. And here you can see that there's something going on that is actually interfering with the activity of this drug, because you can see it's reaching its high concentration, but when it reaches a certain concentration, there is something over here that's interfering. And so instead of getting a nice circle where that drug diffused out, something is interfering with its activity over there. Some common things that you can find that'll interfere with antibiotics are antacids, but also, as some people have found, birth control pills. So you're taking birth control pills, you get a bacterial infection, so you take your antibacterial, and you end up with a parasite for the next 18 years and 9 months of your life. The last thing that we want to consider is something that's known as selective toxicity and efficacy. And if you look at this graph, what you can see here is we're looking at different people's opinions on what they expect antibiotics to be used for. And you can see that there are some distinct differences in people's opinion. And if we look at this, we can see that people on Medicaid have a very different opinion. They want their doctor to basically give them antibiotics because they think that one is needed. And if you don't get it, you take your kid to a different doctor. And this is really a scary kind of thing to think about, that over time we see this massive increase in this opinion from people that if my doctor doesn't give me an antibiotic when I go there because my kid has the flu, then I'm going to go to another doctor who is going to give them an antibiotic. The problem is antibiotics are used to treat living organisms, and the flu is caused by a virus. Antibiotics have no effect on the viral infection, and if anything, they're going to result in giving you diarrhea because they are going to clear out the infection or clear out the bacteria that is in your intestine and not get rid of the infection that is causing you to be sick. So when we look at this idea, people need to think about what's going on with what's known as selective toxicity, and that's the antimicrobial drug should really kill or inhibit the microbe without causing damage to the host tissue. So it is toxic, but it is selective for that particular kind of pathogen that's causing the disease. And when we look at efficacy, there you're looking at basically cost-benefit analysis. And a doctor or somebody who is writing a prescription needs to decide, is the possible damage that could occur from the use of this chemotherapeutic drug worth it? Is it actually going to be worth getting rid of that infection? And so to put this sort of in perspective, think about if you have gangrene and your leg is swelling up. We want to give you an antibiotic to stop that because otherwise we're going to have to amputate. So is it worth possibly losing your kidneys because you're getting a drug to treat the infection, or do we have to amputate and you lose your leg? And so again, this idea of cost-benefit analysis. And when you look at this, in order to come to sort of a good decision about it, what you can actually consider is what's known as the therapeutic index. And this is the ratio of the dose of the drug that is actually toxic to a human versus the minimum effective therapeutic dose. And so if you look at this thing, we want to have a therapeutic index that is really, really large. And if you think about this, if you've got something where you're going to come out with a therapeutic index of 1, that's going to be a risky choice. Yeah, it's going to be the minimum effective dose, and it gets rid of the pathogen that's causing the disease, but it can also be the toxic dose if you're close to a TI of 1. And so we really want to have a therapeutic index that is a very large number to indicate that it's going to require a larger dose that might possibly kill the person, whereas a smaller dose is going to be effective at getting rid of the pathogen there. And so when we look at this, we can use this to sort of gauge the side effects or the damage that might occur as a result of taking this drug to treat the infection. And in order to do this, well, how do you find what is the minimum effective dose? Well, we can come in and do a variety of things, and one of them is what's known as a dilution test. And this can be used to determine what's known as the MIC, or the minimal inhibitory concentration of that. And what we see here is an experiment where somebody has set up a series of tubes here with broth, and they've added a culture containing your cells into it, and each of these tubes has different amounts of a drug. And so if you look, we go from 2 up to 32 micrograms per mil, so we're increasing the concentration of the drug. And then you incubate your tubes overnight and you monitor them. And if you look at this, you can see there is turbidity in these first set of tubes. So we know we did not reach the minimum concentration to inhibit the growth of it. But once we hit the 8 micrograms per mil, we've hit the MIC, and those cells are inhibited from growing. If you go up to a higher concentration, they're still going to be inhibited there. So we know at least 8 micrograms per mil of the drug is needed to inhibit growth. And that's going to be useful because now you can think about, okay, what's the size of a person and how much drug do we need to administer to at least reach 8 micrograms per mil in their bloodstream, because we know that's going to be inhibitory for the growth of this bacteria there. But we haven't reached the point where we're actually killing off the cells necessarily. You might be at what's called the minimal bactericidal concentration, but we can't tell just from this first experiment right here. In order to identify that and to get the dose that's necessary to kill, we're going to take these cells where you actually have your clear broth. You don't see any evidence of growth, but there is the potential that you could still have some cells in there that were inhibited from growing, but they weren't killed off. And you take some of the culture out of this and you put it onto some fresh culture media. In this case, we've got some agar plates, but notice this is drug-free media. And if you look at the results right here for this particular one, it was a clear tube, but you can see we do have bacteria colonies growing on our drug-free media. And so we know we hit the MIC and inhibited the growth, but we haven't hit the minimal bactericidal concentration, or the MBC. The MBC in this one is going to be with our 16 micrograms per mil, because you can see there's no colonies growing there. So our MIC to inhibit growth is 8 micrograms per mil, and our MBC is 16 micrograms per mil in order to inhibit the growth and also kill off those cells. There is some other methods that are out there. Our Kirby-Bauer disc diffusion test that we showed you some example in the lab, where you're looking at your size of your zone of inhibition right here, and Kirby-Bauer actually did these dilution tests, and then were able to figure out the concentration of the drugs that was necessary to kill it. And when we're looking at this, again, when you're looking at it, the size can be indicative of how well the chemical diffuses throughout the media there. But Kirby-Bauer actually standardized this test. And so when we look at this, this can be used to determine whether or not your cells are sensitive or resistant to the drug based on the size of the zone inhibition. And then the last example over here is what's known as the E-test. And here you're looking at this strip where you've got a gradient of antibiotic that is predefined on this media right here. And so if we look at our E-test right here, we can say way down there, there is our MIC. It looks like right around 1.5, whatever the units are on this particular scale. In the next video, we're going to start to take a look at the different types of drugs that are out there and look at their specific mode of action and go through all the different kinds of examples here.